Hello and welcome to Eppendorf Lab Channel, your home for experts and knowledge live. I'm Matthew Yurkevich, a marketing manager for Eppendorf. We're all thrilled to have you joining us for our latest webinar entitled Enhancing Research, Integrating Toxometris.ai's in silico capabilities with eLabJournal. In this session, our speakers will offer insights into using in silico methods for predictive toxicology and drug development, as well as address challenges and solutions, highlighting their significance in advancing drug discovery and safety assessment. This will be a joint presentation from two experts. I'm excited to welcome Zareh Zarabian, head of eLabNext in the Eppendorf Group, and Nelly Babayan, Chief Scientific Officer for Toxometris.ai. Now, before they get started, I would like to direct your attention to the interactive portion of Eppendorf Lab Channel. If you look below the video feed, you will see a short form for submitting questions. After the presentation is completed, we will have a question and answer period where Z and Nelly can address questions that you have submitted. If we don't have time to get to every question live, don't worry. The experts will reply via email after the event. Now, without further ado, I'd like to hand over the reins to Z and Nelly. The stage is yours.
Thank you very much for having us. Thanks everyone for joining. In today's presentation, I'll give you a quick introduction into digital lab platforms and its integration into Toxometris.ai and how this will help with the speed of drug discovery. And then I will pass it on over to Nelly to give you a little bit of more insight. So thank you very much once again for joining, and we're very excited to have this webinar with you, and hopefully we'll have some great conversations afterwards. In today's agenda, in my portion of the presentation, I will talk about a little bit about the eLabNext ecosystem and what makes it special, and especially in today's world of digital transformations. Then we'll talk about power of integrations, of course, with the Toxometris as being the focal point, and we'll go from there. So, you know, the end goal is always to find the drugs that are going to help us with therapeutics. This is the beginning of any laboratory research. The goal is to get to this point. But to get to this point, the lab has to go through a series of transformations in order for them to do it in a sustainable way. So to look into this a bit further, let's look at some of these slides over here. So we do have a saying, we say your sample, your lab, our passion. And the reason why we say this is because we believe that everything starts with a sample. This sample can be blood samples, tissue samples, cell lines, tumor samples, whatever the research is on. It all starts with people doing research and development in the lab and collecting samples. And this is happening in a manual way in many labs, which is not very sustainable. There's a lot of misplacement of samples. There's a lot of issues of collecting data and accessing the data. So this path towards drug discovery, it all starts from this transition from manual sample management and lab operations and transitioning over to digital sample management and lab operations. This could be done through a simple means like getting the safe code tubes, tubes that are already barcoded, collecting the data and the metadata that's associated with these samples, all the way to collecting all of the instrumentation data that you get from analyzing these samples. And this brings us over to more organized sample management and overall organized lab operations, which then essentially brings us over to sustainable sample tracking and lab operations. When we use the word sustainable, this is not just the buzzword that gets thrown around in the industry these days. This is really about building an ecosystem that is going to last a lifetime. And having a standardized data management, and not just sample management, but everything else that comes along with it, from the experimentation, collection of instrument data, and so on, will allow labs to mine the data in the right way in order to get to that drug discovery much faster. So in this process of lab digitization and digital transformation, the whole goal is to get to data connectivity in a sustainable way. And in order to get to that, there are key pillars that we stand on. And these pillars are titled as accessibility. So this is not just accessing data remotely from home or whatsoever, but it's about accessing large sets of data. So for example, let's say you have 15,000 compounds in your laboratory. You're building a large molecule compound library. You have a biobank of information. All of these have, let's say all of these compounds have 50 metadata fields, and then each one of these metadata fields has attachments, CSV files, or many other files that have hundreds and thousands of other metadata fields. Well, then this is a deep data lake that needs to be mined. And it's about having a system that is so indexed and so standardized that it allows you to access that information very easily through the means of the software that you are using, the digital lab platform that you are using. The other one is, of course, efficiency. So whether it is using automation tools, whether it is using mobile apps that can help you with the lab operations, oftentimes we only think about kind of after all of the lab work, the wet lab work has been done, what it's like to, let's say, be more efficient. But all of that work that the scientists do in the lab, I call them, the folks that are in the trenches doing all of this hard work. It's important for them to be efficient because when things get misplaced, when things cannot be accessed, it all adds up hours upon hours towards that finding those therapeutics for the patients. So efficiency is another key pillar of digital transformation. Then there is, of course, security and compliance. And I'll talk a little bit about this in the next slide. But if you are prepping to go into the clinical stage, everything has to be compliant, whether it is HIPAA, GDPR, and so on. And of course, if you're working with patient data, this needs to be secure as well. Then, of course, there is data analysis, which Toxometris.ai will talk about a little bit more. But, traditionally, data analysis was not part of the conversation, let's say 5-6 years ago, when it came to lab operations, because all of the data was initially collected and then sent over to a CRO for data analysis. But now we have tools at our fingertips that allows us to analyze the data in real time, which allows us to pivot the research towards the right direction as needed and not waste years upon collecting data and then years upon analyzing that data. And this, to me, is a complete game changer. This is completely revolutionary as to what it means to drug discovery, and Toxometris will talk about that a little bit more very, very soon. And lastly, it's instrument monitoring and connectivity. This can mean many different things. It can be as simple as, let's say, monitoring temperature and humidity of your freezers and incubators and knowing the viability of your samples, all the way to being able to control robots from a digital lab platform, being able to push and pull data from a digital lab platform to equipment and back to the platform, all the way to pushing it over to hospital e-health systems and so on, and being able to analyze that moving forward. So this whole ecosystem is being powered by the API and the SDK of eLabNext, which then opens up a marketplace where it allows you to integrate these additional tools that can do some of that work that eLab does not specialize in, one of them being drug discovery. So this is where Toxometris.ai lives within this ecosystem, and we'll talk about that as we go through this presentation. So I hope this slide brings a smile to your face because it is so relatable to folks in the lab. When you look at the person on the top left, this is where we want to be in the lab, right? We want to be very organized, not just with our samples and lab operations, but how we collect the data, how we analyze the data, what kind of predictive modeling do we have to apply to the data that we collect, and so on and on. But oftentimes what happens is we end up being the person in the bottom right, not just with our freezers, but how we actually collect the data and how we analyze the data. When you have Excel files living in 20 different computers on the local server with different names, different folks working with it, when things are not standardized, this can cause a lot of issues. And we see this happening all the time. People going through lawsuits, companies going through lawsuits, companies losing their patents, and so on, because they are not standardized in the work that they do. And this is where we come in. We come in with a digital lab platform that allows you to use this digital tool in order to be physically more organized in how you collect the data and so on. So when we look at the drug discovery and development timeline, we see that this is something that takes a very long time. So this allows us to kind of come up with these questions where we say, okay, how can we cut the drug discovery stage when you have 15,000 compounds at best? And how do you pick the right compounds in order for you to do your clinical trials? How does eLabNext help with this? This is another question. Well, we are not in the drug discovery, but we are in the previous stages that allows you to collect the data in the right way to get you ready to be AI ready in order for you to be drug discovery ready. And this brings us to the last question is, how does Toxometris.ai help with this? And as we go through the rest of this presentation, you will be able to find the answers to these questions. So before we go any further, just a little bit of background information that is important in drug discoveries. You want to use platforms that are ISO certified. So eLabNext is an ISO certified system, as you can see, and we are FDA compliant, ISO certified, GXP compliant, GDPR compliant, and HIPAA compliant. So when you're working with patient data, when you have to store, you have to be CLIA compliant as well when you're storing patient data, and having an ecosystem that you can trust based on these certifications is extremely important. So, drug discovery can come in many different ways, and the research that you do in the lab can be very different as well as you go from lab to lab. And it's important to use a system that is not only configurable, but also flexible. So, for example, eLabNext, and one of the reasons why we have all of these names over here is to show you how flexible and configurable the system really is. Whether it is a small biotech company with 5 people, or a big pharma company like Bayer with hundreds of people, or like Institute Pasteur that has 5,000 users of eLab, it does not matter. The whole idea here is that the system can adapt to the specific workflows. Can the system handle and all of this data that's being generated? And then can the system be mined by solutions like Toxometris.ai to further the research into the drug discovery stage? And the answer is yes. That's exactly the kind of solutions that we provide. And the whole idea here is that to have the connectivity and be able to grow with the system as the research and development operations change and morph through time. And what allows us to do this is having a holistic solution. So instead of giving you pieces to the puzzle, we say that we solve the whole puzzle. And the idea here is that you are not getting disconnected data sources and silos, but instead you are connecting everything. You are not using one-dimensional solutions, just small pieces of the puzzle, but you are using the whole puzzle altogether. And the current available tools are not future ready when they are only focusing on one aspect of the lab work. And this is where the eLab Next ecosystem comes in, allowing you to be future proof and allowing you to stay away from one-trick ponies and focus on the whole puzzle and have the whole solution in front of you. So to drive this message a bit further, let's look at this slide over here. So a little bit of a history lesson for the folks that are joining in. In the 1800s, of course, we had the cable landline. Maybe some of you have used this. I know I have. They were actually kind of fun as a kid to play with. And these are as old as physical notebooks. I'm sure you've seen physical notebooks in your lab. Maybe you still use it yourself to take some notes. But there was definitely a change that happened in the 1990s when we transitioned into the mobile phones. Oh, and then this was the time when electronic lab notebooks were introduced. There was Microsoft Office where people started taking notes in Word documents using Excel and some of the other tools that were available at the time. But those were not very sustainable because labs grew out of these systems very quickly. They were not updated in a sustainable way, and so on. So in 2000s, there was another digital revolution that happened where the establishment of the smartphone happened in 2007, where let's say you no longer needed a calculator, you no longer needed an additional GPS. Everything was in an app format within that mobile phone. And this is where we came in with eLab Next, providing labs with a digital lab platform, a lab operating system, where we give you the foundational needs of the lab. But instead of us creating tools like Toxometris.ai, well, we're not the specialists at that. Nelly and Zavin are. So we essentially integrate with them in the form of an app within our platform. So we do not reinvent the wheel, but we extend and expand the capabilities of this ecosystem that we've been able to build. So to recap, the key modules of eLab are eLabJournal, eLab Inventory, and eLab Protocols. And this allows you to do all kinds of things in the lab, from sample management to supply and ordering, automation of your robotics, electronic lab notebook, management of your SOPs, and so on. And with the power of the API and SDK in the marketplace, this is able to get connected to other systems, other platforms, and it extends the capabilities of the lab. And here you can see some of the images of what the UI looks like, but essentially the end goal being towards drug discovery. As we provide this ecosystem with all of these data points, then companies like Toxometris.ai can mine that and take you to the next stage in drug discovery. And to help you guys to think about this in a more holistic way, and of course, I won't read through all of these questions over here. You guys can come back and take a look at these questions and think about them. But I really welcome folks to look at some of these right now together, where oftentimes when we digitize our lab, when we digitize the workflow that we do, or if we're about to start using an AI tool, we don't think about the long-term ripple effects. We essentially only think about what we need in that right moment. Okay, here's a tool, let's quickly use it and move on to the next thing. But I really welcome folks to think about the long-term effects. So some things that we don't even think about. So for example, we talked about the deep data lake. When you have 200,000 samples, and all of these samples have metadata fields upon metadata fields, now we will see how Toxometris AI will be able to mine that data. But what about any other AI tools that we don't even have access to yet, and they are currently in development. All of this data that you are collecting, if you don't do it in a standardized way, you won't be able to reap the benefits of it in the future. So the more organized and standardized you are now, the better off you will be in the future. So what are the long-term goals of your data collection is something that should be asked. Another example is, okay, all of these hundreds and thousands of compounds that you're collecting, you're storing them in, freezers and fridges and, all of these places. When you know where these things are located, when you can access them and so on, you're spending less time looking for them. But most importantly, you're not opening and closing freezer doors left and right and letting out the energy out, and this can have a ripple effect in a large corporation and big pharma when you have hundreds of freezers. When all of them are opening and closing every day, this can rack up the bill within the building. This might seem like a trivial thing, but this really ends up having a huge ripple effect and huge savings for organizations. So I'll leave it there with these questions, but I do welcome everyone to kind of come back into the slide and look at these questions and think about what they're doing in the lab that can be optimized. Because having the digital tools that you use in the lab can really save you a lot of time and a lot of money, not just in the short term, but in the long term. And I do want to mention that it is a tangible dream. And what I mean by that is whether you have already digitized a lot of your work or you're just starting out, maybe you have legacy data of 30 years of data being collected in Excels and local servers and so on. We specialize in helping labs to transition to a digital space. So we will be able to help you with that. Do not worry. While you should be cautious and very methodical, it doesn't mean that it's not doable. And we have a lot of experience with helping labs with this. So to recap, eLab is the brand. The modules within it is eLabJournal, eLab Inventory, and eLab Protocols. And then the extension that we will be talking about today is Toxometris.ai, a powerful AI software that helps you with drug discovery. And within the ecosystem of eLab, to help you guys to visualize this even further, you see here on the right the Toxometris AI logo. But when you look at the overall system of your lab operations, your goal towards drug discovery, your core data models, digital strategy, business strategy, and then you connect that with the API and SDK tools that eLab provides, and you couple that with its core modules of eLabJournal, eLab Inventory, eLab Protocols, and maybe other tools that eLab provides. But then you can look into the overall ecosystem and in the overall industry and say, hey, we also want to use other tools that are useful for us. And this is where this comes in. And you are able to connect other tools like, let's say, for image, if you're doing microscopy work, you can use Modicus Prime for your computer vision analysis. If you're monitoring the temperature of your freezers and so on, you can use Elemental Machines and Visionize. If you're doing multi-omics analysis, you can use Immunomind. And the list goes on and on. So this is the ecosystem that we provide, and we believe that having this setup where you have foundational data management, in addition to having these extensions and other tools that focus on specific niche things that you need, is what is going to help labs to propel forward and be competitive in the industry with the rise of AI tools that we see. So last but not least, I do want to give thanks to the team that's currently the team that I'm speaking of that's here in Boston. And if anybody that wants to reach out to any of us here within eLab, we're more than happy to set up meetings with you, 15-minute calls, 30-minute calls to discuss how we can help and how Toxometris AI can help with any lab needs that you might have. So I'll end it here. Thank you very much, and I'll pass it on over to Nelly to continue with the presentation of Toxometris AI.
Thanks, Dare. It was very interesting. Thanks for your kind introduction. First, I'm going to speak a little about in silico world to understand how virtual predictions are changing the game in toxicology and drug development. Next up, we will talk about ADMET properties to understand how chemicals interact with the human body and what kind of models are used to predict those properties. And to bring all this to life, we will cap off our agenda with a demonstration to see these predictions in action. But let me start with a quite optimistic slide, which represents the current paradigm of safety toxicology. So historically and up to now, an extensive amount of experiments has been performed in vivo using animals to assess the potential effects of chemicals in humans. And there is an unshakable truth that we obviously know how to make this world safer, primarily for rats and mice, but not for humans. So ethical, economical, and human relevance issues forced to develop two main alternatives to animal studies, which are in vitro, using isolated cells or tissues, and in silico, which is a computer modeling. Today, these approaches are supportive for decision-making organizations, but in the future, they are likely to replace animal studies, and this future is not far from us. So almost all in silico predictions are based on QSAR models. So they are quantitative, relying on statistically derived models, while SAR models are qualitative, based on expert rule-based approaches. And the general assumption of those QSAR models is that similar molecules exhibit similar properties. But what is chemical similarity? What kind of similarity of chemicals do we need to address? For example, the Tanimoto coefficient is one of the most used measures to estimate the structural similarity of chemicals. But as you can see, chemicals which have high Tanimoto similarity in real life exhibit different biological activities. One is genotoxic and the other one is not. And here on the right, you can see highly dissimilar chemicals that exhibit mutagenic effects. And by the way, they both have epoxy groups, indicating that not only the effect, but also the mechanism of the effect may be the same in these compounds. So general assumptions should be approached with caution, and more complicated relationships should be estimated by today's QSAR models. As I have mentioned, two main approaches are used for in silico prediction, statistical based, which are comparably faster to construct. Here, both relatively simple models such as random forest and support vector machines, as well as more complex ones such as graph neural networks or even large language models can be used. The second approach is SAR, expert rule-based model, which are time-consuming to build, but since they are knowledge-based, they are supported by mechanistic information of the predicted effect. These models face a significant challenge. While they accurately predict positive outcomes, their performance for negative predictions is often low. This is because the absence of knowledge does not imply the absence of the effect. There is also a third widely used approach, which is read across, which is not actually a prediction model, but a searching approach to find similar molecules with known experimental data and make estimations based on properties of these analogs. But before using any kind of in silico models, it is important to know to which chemicals it is applicable. The applicability space of most QSAR models typically includes organic chemicals with a molecular weight of less than 1,000 Dalton. With special attention, polysaccharides, organometallics, and mixtures properties can be also predicted. However, QSAR models are unable to make predictions for polymers and inorganic compounds. And additionally, distinguishing between stereoisomers remains a significant challenge for QSAR modeling. It is also important to be ready that all in silico predictions share a common challenge. Actually, they predict the probability of the effect or property. Whether that probability is high or low, it introduces an inherent uncertainty, making it impossible to obtain the exact answer to a given question. And now you may question, what's the point of using in silico predictions? Is there a meaning? But the answer is yes, and the following are the solutions. First is ranking. If you work with a large quantity of chemicals, it's beneficial to conduct an initial ranking based on the properties of interest. Select the most promising compounds and only then perform wet lab experimentation for these chemicals. But in case if you have just one compound, it can be screened towards different properties and the most problematic properties will be revealed. So in this case, the wet lab experiment will be started from this problematic property. If an experiment confirms bad prediction, at least this discovery will happen in the early stage of studies. So this approach will optimize the efficiency of wet lab experimentation in terms of time and cost. The second solution is explaining, particularly the mechanistic interpretation of predictions provide a degree of assurance in the decision-making process, offering a sense of confidence and certainty. This approach is viable when dealing with a limited number of compounds. Because there is a need to invest time to delve into the mechanistic evidence of predictions for each individual compound. Another aspect to consider when using in silico predictions is the applicability domain of the models. Applicability domain is the space of chemicals within which the model's predictions are reliable. So in case of knowledge-based models, the applicability domain is usually small and restricted with a limited amount of knowledge-based molecular substructures associated with the effect. But in case of statistical QSAR models, the applicability domain is comparatively large and is constrained by the training dataset. So it is obvious that the broader the chemical diversity of the training set, the greater the applicability domain of the model. But not only the diversity, but also the size of the training set contribute to increase the accuracy in predictions. The problem here is that it is very expensive to obtain as much experimental data as possible. So here, two solutions address these issues. First is a fine-tuning approach. This approach is viable when experimental data is accessible for a subset of compounds within a specific family, enabling the adjustment of the general model to adapt to the unique characteristics of these compounds. And the second approach is active learning, which is employed in situations where there is limited or non-existing experimental data available. In such cases, the model selects a small subset of compounds for wet lab experimentation. By incorporating these additional data points, the model enhances its performance specifically for that family of compounds, and this approach aims to achieve maximum performance using minimal experimental data. So having this background information in mind about in silico predictions, now Let me switch to the second section of our agenda, particularly what kind of effects and properties can be predicted to understand the possible interactions of chemicals with the human body. So this is ADMET properties, which is an abbreviation from absorption. It is to understand how the chemical enters the body. This stands for the distribution of chemicals in different tissues. Metabolism is a biotransformation of the chemicals. Excretion, actually, when speaking about in silico prediction or excretion, we consider the rate at which a substance is eliminated from the body through specific pathways. And finally, toxicity, which refers to a different adverse effect caused by the chemical. So in predicted ADMET properties, Toxometris.ai applies all the above mentioned approaches, statistical, where cutting-edge AI/ML models are used, rule-based methods, and read-across techniques. Of course, the number of properties may seem unlimited, but here you can see the key ADMET properties we are focused on, which are fair enough to understand the potential effects of the chemicals on the beginning of the journey. In addition to individual models that predict specific endpoints, we provide a ranking function capable of screening a large number of chemicals across various endpoints of interest. This function assigns a single score to each molecule, facilitating the ranking of compounds from the most promising to the least. Using this feature, you can focus your efforts towards the most promising compounds and avoid wasting time and resources on less favorable ones. In our predictions, we use ensemble models, which make a consensus prediction taken from a combination of multiple individual models. Individual models have been selected in a way that they can operate on different types of molecular representations. The reason is that each type of molecular representation exhibits different types of information about the same molecule. And depending on the endpoint, the final prediction may be positive or negative, as in the case of genotoxicity, which is a classification task, or in case of solubility, which is a regression task, we predict the value, which is logarithm of solubility. And here you can see an example of the report file, which has been developed in accordance to regulatory requirements. Particularly, it consists of 3 blocks: statistical, rule-based, and read-across. Statistical-based approach is based on our ensemble model that I mentioned above. Here you can see the prediction outcome of each model with confidence. So for this molecule, the prediction is non-mutagenic with high confidence from each model, and based on this prediction, you will see the prediction results in the reliability of the prediction. In the rule-based block, you will find the results for the estimated top 6 positive or negative structural alerts, which enhance the explainability of the prediction. And finally, you will see the experimental values of 3 nearest neighbors from the training set. So this was the report you could obtain for each endpoint, but when the scoring function is applied and the risk score is calculated, the final list of ranked compounds is constructed. So the most promising compound appears at the top of the list, while the toxic ones at the end. And here is an example of such a study performed for one of our customers, and our report recommended started the wet lab experiment from the compound for number 5. Additionally, the scoring function can be used to rank compounds based on their physical, chemical, and medicinal chemistry properties, enhancing the ranking list obtained solely from ADMET properties. The important point to note when using any kind of in silico predictions is that general models developed based on even large and diverse data sets can suffer from overgeneralization and may perform poorly on specific families of compounds. In such cases, we offer the fine-tuning approach, where general models are adjusted to perform better for these specific families of compounds. This adjustment helps to better capture the complexities of specific chemical structures and, as a result, enhance the predictive accuracy. But as I've mentioned, the fine-tuning is possible to make when experimental data is accessible for a subset of compounds within a specific family. If there is limited or non-existing experimental data available for a particular family of compounds, we can apply an active learning approach. This approach allows us to improve the prediction performance of the model by incorporating experimental values for the chemicals of interest, which can constitute from 5 to 20% of the training data. This type of model can be custom developed, ensuring it is optimized as the best available model for the targeted family of chemicals. So let me switch to the demonstration of the platform, which is already accessible through the eLabJournal. And I'll hand over the floor to Zaven for the platform demonstration.
Thank you, Nelly. Just a moment, I'll share my screen. Okay, here we can see the eLabJournal interface. So we go to the inventory. Here is a sample list where our test chemicals are stored. For example, we can choose one of our chemicals, caffeine. Then we click on Send to Toxometris button. And this chemical automatically is transferred to the portal Toxometris.ai. And if you have an account there, you can directly push login, and you will see the Smalls formula of the compound appeared in the new test form. So then we simply click Next, and we choose the test of the interest. For example, the Ames mutagenicity test. We can also select multiple tests, but for demo, I think it's enough. So then we click Next, and then click on Submit. Now we go to Perform Test section, and we can see that this Ames mutagenicity test is currently in the process. And we refresh, and it's already ready. Then we can click on the View button and see the final report. Final report for the specific compound. And we can see here that the caffeine is predicted as a non-mutagenic, which is very good because it's one of my favorite drinks. So here we can see the positive and negative alerts, and also read across study with 3 similar compounds. So all they are non-mutagenic. I think that's it. Nelly, do you have something to add?
No, thank you for the demonstration. I think we can switch to the questions, maybe.
Great. Yeah, actually, I do have some questions. Well, first of all, I'm so happy that caffeine is safe. I've already had 2 espressos, so that's great news that we don't have to worry about it. My first question is to Nelly and Zaven. Maybe Nelly, you can help with answering this. What is the percentage of error possible using this tool? And maybe, what are the best ways to deal with that, especially when it comes to distrust in quality of data that they have collected in the first place? How do you guys deal with that?
Yeah, thanks. It's a very practical question. The percentage of error actually can be reflected in the accuracy value available for each in silico models. Usually, you can find these values in QMRFs, which in case of Toxometris are available on the portal. So there you can find comprehensive information about the model, including not only the accuracy but also sensitivity, which indicates how well the model predicts positive values, and its specificity, which shows how good it in prediction of negative values. So, for example, in case of the same AIMS test, which is a genotoxicity prediction model, the accuracy is around 84%. So an error rate is around 16%. Yes, but it is important to know that interlaboratory reproducibility of this test is around actually 86%. So an 84% accuracy for in silico prediction represent the peak possible accuracy for this test. And
That's great.
yeah, of course, the applicability domain plays a crucial role here, as model can only provide the mentioned accuracy for molecules with structures close to the training data. Yes, and the quality of data is, of course, a cornerstone for in silico models, and it's better to use less but high quality data. So we construct our models based on experimental data which have been performed in accordance to guidelines or standard protocols, and this is the only way to build an accurate model.
That's great. Yeah, that makes a lot of sense. Thanks for that insight. And when it comes to, let's say, your competitors and yourself, is there a high computing cost when it comes to running these scripts? Can you talk more about that?
Uh, well, yeah, thanks for the question. Usually, the training process is computationally costly everywhere, as it uses both CPU and GPU time and involves data processing. But however, the inference part, as you could see with the demonstration, where actual predictions are made based on user smiles, is not very resource consuming. So predictions for a relatively small number of compounds usually take only a few seconds to complete.
That's good to know. And okay, my last question here is, and it's a bit of a more abstract question, so maybe we can have a discussion on it. What do you think will the relationship be between AI and actual clinical trials moving forward? What do you anticipate the next several years to look like? And do you have any predictions on what 10 years from now or 20 years would be like with tools like Toxometris.ai kind of taking over these processes?
Yeah, thanks. AI is everywhere now, and AI is a very powerful tool in various industries today. And we are witnesses of deep integration of AI with the pharmaceutical industry in recent years. And as you could see from the slide, next 10 or 20 years, it can replace at least some animal experimentations, but using this kind of screening models, so AI can rapidly screen potential drug candidates, identify safety concerns, and optimize dosing regimens. So this enables researchers to prioritize compounds with favorable ADMET or other profiles, and streamline their drug development process. And of course, it reduces the risk of adverse effects during clinical trials. Yeah.
Right. Yeah, I'm very excited for it. I think, the industry needs a couple of success stories because many companies are already using AI and have been using AI for some time, but not from end to end, right? AI comes in different portions of the lab and optimizes it. But once we have that success story from beginning to end where AI has helped to make it more efficient, I think that success story is what will help to change the expectations of the speed at which we find the therapeutics that we need. And I believe that Toxometris AI will play a huge role in that, and I hope it does. So thanks for participating in this webinar. Big thanks to the Eppendorf Lab channel for organizing this, always bringing innovative technologies to the forefront. So thank you very much, and I hope that the folks that are tuning in will enjoy this conversation and reach out to all of us to ask any further questions that they might have. Thanks, everyone.
Thank you.
Thank you.
Thank you for joining us today, and we look forward to seeing you all at our next broadcast. You could go to eppendorf.com/labchannel to find out all the details and save the date in your calendar. If you missed out on part of this broadcast or any of our past events, you can also view all our previous episodes on demand at eppendorf.com/labchannel. Watch them all on your own time and use the links to contact the experts with any questions, comments, or reactions. On behalf of Z, Nelly, and everyone else here at Eppendorf Lab Channel, thank you for taking the time to join us, and until we see you again, stay safe and have a wonderful day.